All right, thank you everyone for joining today's webinar. Today's talk is called Create Your Own InCELL Pulse Cellular Target Engagement Assays. My name is Dana Haley Vicente. I'm the product manager at DiscoverX. Just wanted to go through a few housekeeping items before we get started. Today's webinar is recorded, and shortly after the webinar, before Friday, you should receive the slides and the recording to download. During the session or towards the end, please use the chat or the Q&A pull downs in WebEx, the upper right-hand corner, to ask any questions. I'll be reading these to our speaker today, and we'll get back to you. If your question is not answered due to time, we will get back to you via email. All right, today's speaker is Dr. Daniel Trieber. He is our Chief Technology Officer and the main lead developer of this new technology called InCELL Pulse for compound target engagement. And with that, I'd like to introduce to you Dr. Trieber.
Thank you very much, Dana, and thanks everyone for joining today's webinar. I'd like to tell you about some new technology that my team has developed that hopefully addresses some of the unmet needs you have on your teams. So the agenda will be as follows. I'll give a brief overview of what cellular target engagement is and why it's important. And then I will describe some tools that we've developed at DiscoverX to create your own InCELL Pulse target engagement assays. I'll describe how the assay development works, what the experiment design and workflow is, and then show some case studies and describe some of the applications of this technology. And we'll finish up with a brief summary and Q&A session. Cellular target engagement. It's really a core concept in pharmacology. Small molecule drugs work by acting on protein targets to elicit hopefully a desirable phenotype, and so this is really a key underpinning in pharmacology, and it's actually quite difficult to get at. Usually the assays that measure cellular target engagement are somewhat indirect, measure a functional readout or a phenotypic readout, and wouldn't it be nice to figure out if a compound is directly engaging the target in the cellular milieu? So what is cellular target engagement? It's a very simple concept. A small molecule enters a cell and engages the target of interest. And like I said earlier, this is a difficult thing to measure. Historically, it's been a very difficult thing to measure directly and has often relied on functional or phenotypic indirect type assays. So in this cartoon, we have a depiction of a cell. The small molecule enters the cell and binds to its protein target. This is the bound state, and of course there's also going to be some of this target in the unbound state. And so where in the drug discovery continuum is target engagement important? When do we need to measure this? And so it's actually important for a couple of different drug discovery paradigms. So target-specific approaches that are very common these days, where we start with a target-specific biochemical type screen and identify hits. And then, of course, the next step is to prove that these compounds have activity, target-specific activity in cellular assays. And this is generally done by functional assays or, in some cases, more indirect phenotypic assays. So this is a very nice place where a direct target engagement assay would be applicable. And then, of course, once that's established, compounds can be tested in more complex cellular and animal disease models, and this will help to validate the target and to evaluate the drug-like properties of a particular compound. Now, a target engagement approach can also be applied in a phenotypic screening-based approach where a particular phenotypic assay, perhaps a cell-based assay, is screened against a compound library, and certain hits are identified that give the desired phenotype, and then there are some detailed and very difficult, actually, target deconvolution studies. What is the target of my compound that's giving this effect? And that generally will deliver some good hypotheses, but then those hypotheses need to be tested and validated by proving that the compound actually engages the target in the cellular milieu. And then these types of data, if they work out the way that we all hope, will validate a new target. And so these are two key areas where a cellular target engagement assay could be and should be applied. So what are the assay needs out there for the clients that we interact with for these cell-based target engagement assays? What folks are usually interested in is they want to ensure that their compounds get into cells and engage the target of interest, and they want to be able to rank order how potent those compounds are against the target. And they want to, like I said earlier in this phenotypic screening description, they want to confirm target engagement for the target that they believe is responsible for the phenotype of the cells they've treated. And so these are the questions that they're addressing. Is my compound cell penetrant? Does my compound bind to its intended target? Or maybe also to some off targets that were identified in other screens? And how potent is this engagement? And so ideally, the assay would fulfill the following requirements: quantitative, simple to use, HTS compatible, and low cost. And we also would like to provide a generic assay format with a generic readout that's applicable across diverse cellular targets, and we'd really like to reduce the requirement for custom reagents. And of course, any assay like this should provide accurate rank order inhibitor potencies. So, like I was saying earlier, measuring direct target engagement has been a real challenge in pharmacology. Most cell-based assays rely on some kind of a functional readout or a phenotypic readout, for example, an autophos assay for kinases. There was a breakthrough made a few years ago with the cellular thermal shift assay, CETSA, which I'm sure you're familiar with. And this is a very powerful concept of using thermal stability changes induced by ligand binding to actually prove and to demonstrate that compounds interact with targets and cells. And so this CETSA format is highlighted in this cartoon to the right, where if the cells are heated at increasingly high temperatures, the target protein denatures and aggregates. But if a ligand is bound, the temperature required to denature aggregate these proteins gets shifted to the right. And this concept can be exploited to then use an isothermal type approach as a function of the compound concentration to measure inhibitor potencies. And this type of assay is generally read out using an ELISA method or a Western blot method as shown in this example. So because this readout is cumbersome, it's fairly slow and low throughput, and of course it requires a custom antibody for each target being evaluated. So we were thinking that we could apply some proprietary DiscoverX technology to this thermal shift assay concept and address some of these bottlenecks. So InCELL Pulse is our solution. This is a new product for DiscoverX, where we've come up with and designed an engineered cellular thermal shift assay that uses core DiscoverX technology. And because of the way the assays are run and read out, it's a very simple and rapid method. It's a generic quantitative method that uses a standard luminescence readout. And these are some model data or real data that we've generated. So you get the TM shift data using a nice homogeneous quantitative readout, and then this can be transferred to isothermal experiments where we measure inhibitor target engagement potencies in the cellular environment. So what are the tools that we provide to create your own target-specific InCELL Pulse assay? So before I get started on exactly what these tools are, I'd like to describe how InCELL Pulse works. So I mentioned earlier that it's underpinned by some DiscoverX proprietary technology. And what is this technology? It's enzyme fragment complementation, EFC technology. So beta-galactosidase can be split into two inactive fragments that complement one another. So it's split into a very small piece, the prolabel tag, which is about 40 amino acids, and a much larger piece called the enzyme acceptor. And when these two pieces come together, beta-gal activity is restored, and with our luminescent substrates, we generate light. Now, what's great is that this small peptide can be appended in frame to a target protein of interest. And that enables the levels of this protein to be assessed using enzyme fragment complementation. And why is this EFC system ideal for InCELL Pulse target engagement assays? Well, first of all, the EPL tag allows for sensitive detection of the properly folded protein, but the EPL tag is also a biophysical tag that senses the folded state of the protein. If the protein is denatured or aggregated, the complementation between EA and the target protein is reduced. And so we can monitor the folded state of the protein using this technology with a very simple readout that doesn't require antibodies or centrifugation steps or any of the complexities of CETSA. In a little bit more detail, here's a little information on the workflow. So we have so it's an engineered system, and we have the EPL tag target protein expressed in a cell, and we treat that cell with a compound or with the DMSO control. The cells go through a brief heat denaturation step in a PCR machine, and if there's no compound binding, of course, at the specified temperature, the target protein denatures and aggregates, and the enzyme fragment complementation is inefficient, and there's not a lot of light generated with the chemiluminescent substrate. However, if compound binding occurs, that protein is stabilized, and that precludes denaturation aggregation. There's still good complementation with the enzyme fragment system, and substrate is turned over and there's a lot of light. So this tag is a very convenient tag for these types of assays. So what do we offer? So we offer a couple of assay-ready kits, but our main offering in this area is what we call the starter kit, the InCELL Pulse Target Engagement Starter Kit. And these provide all the tools for the end user to build their own cellular target engagement assays. And so when this works well, which is often the case, you can create your own user-defined target engagement assay after the cloning steps, of course, in less than 2 weeks. And you have ultimate control over assay development. What constructs do you use? What cell line do you use? And we've actually designed this in such a way that transient transfections work quite well. So there isn't the need to build a stable cell line before trying to develop one of these assays. So what comes in the kit? So one of the key components is this vector kit. So we provide vectors for cloning your target gene in with an N-terminal or a C-terminal EPL fusion. So putting the tag, the complementing tag on either end of the protein. And importantly, we also can include a positive control vector. So this is an assay that we've already developed for MTH1 hydrolase that works really well. And this should be run in parallel just to make sure that everything's working right. It's a positive control. We include enough detection reagents for 400 data points. And we also include plates that are compatible with not only the PCR machine required for the denaturation step, but also for the luminometer, so no plate transfers are required. And we include some control reagents just to make sure that all of the reagents are working well and that the assay is optimized. So now I want to take you through how these assays are built. And so we provide, of course, all the reagents and vectors that I've described on the previous slide. But we also include a very detailed user manual that walks through all the assay development steps, troubleshooting, frequently asked questions, etc. And assay development breaks down into two major phases. So there's the cloning and transfection stage. Then there's three different assay development phases, and I will go through these one at a time. And then there's also at the end, if you've built an assay that you're going to be using quite often, maybe you don't want to do transient transfections every week and you'd like to build a stable cell line, and we provide information about that as well. And the user manual can actually be downloaded free of charge from the website if you're interested, just to see what this process entails in great detail before you decide whether to go in this direction. So now I want to get into, excuse me, some more detail on the experimental design and workflow. So the assay development is in multiple phases, three key phases. The first phase is cloning and transfection. So in the cloning step is really critical. So the goal here is to add the EPL tag to the C-terminus or N-terminus of your target protein of interest. And we provide the vectors for this. And you really want to take some time to decide what constructs you want to put in. So we recommend trying full-length protein, and of course also just a construct encoding the catalytic or compound binding domain that you believe is relevant. And we also suggest trying the pro-label tag on both the C-terminus and N-terminus of any construct that you make. We see that that can make a difference in some cases. And then just a standard transfection of transient transfection of mammalian host cells once you've got these constructs. So you've transfected the cells on day 2, and then the next day you harvest the cells and run the assay, running the assay is very quick. So here's an example of the cloning vectors that we provide. This is the example we're showing here, is one that places the EPL tag on the N-terminus in frame with your target protein, showing the multiple cloning site. We have instructions for how to make sure this is in frame, and we also provide, of course, the vector that will put the EPL tag on the C-terminus of the protein. So after cloning is completed, assay development phase begins. And the first phase of assay development, which is very important, is to optimize. So we have this wonderful EFC-based readout, and there are a couple of variables here that need to be optimized based on how well your protein of interest expresses. We don't want the detection to be too sensitive. We don't want it to be too insensitive. We want to identify conditions that ensure a high signal-to-background ratio for detection of your native properly folded protein. And so these steps are quite simple. The transfected cells are harvested. The assays are assembled and set up in such a way that we're testing multiple conditions for the EFC detection reaction. We perform that InCELL Pulse detection reaction, analyze the data, and then based on these data, we select the optimal EFC detection conditions. And this is a very simple process outlined hopefully clearly in the manual, and once these detection conditions are identified, they can be held constant going forward. Please do not skip this step. Phase 2 assay development starts getting into the real meat of the assay. This is the thermal denaturation assay. We want to show that a known ligand can shift the TM, the apparent TM of the target protein, and then we can choose appropriate temperatures to move forward for assay development. So in this model data, what we can see is as we heat the cells to increasing temperatures from 37 to 60 degrees, the amount of EFC signal we get decreases very steeply as a function of temperature, and that's because the target protein is denaturing, aggregating, and the enzyme fragment complementation becomes less and less efficient. So it's a very convenient readout of that denaturation aggregation process. When a compound that engages the target binds, that TM shift occurs. So the target protein will still denature as a function of temperature, but at higher temperatures. So this horizontal arrow indicates the TM shift. And then at any given temperature where this TM shift has occurred, there's an actual assay window. So at 50 degrees in this example, there's an assay window. So if we were to titrate the compound in, we would start at this much signal, and then as a function of compound concentration, get up to this much signal. So in this experiment, we really want to show that a known potent inhibitor, hopefully with known cellular activity, but ones with biochemical activity can be fine as well, that we get this TM shift. And there are also some important control experiments that we highly recommend to run to make sure the compound isn't having effects on the steady state protein level or on the detection reaction. And those are described in detail in the user manual. So what are the protocol steps for this experiment? So very similar to the previous phase 1 assay development protocol, with a couple of exceptions. So there's a control inhibitor that needs to be prepared. The transfected cells are treated with that control inhibitor or the DMSO controls. The assay is set up and incubated with the compound, say 37 degrees for 30 to 60 minutes. And then we do the denaturation step in a thermocycler. So the cells are in suspension at this point with the compound. It's really nice to have a PCR machine that will do a thermal gradient so that one plate can be added to one PCR machine and we can get denaturation data from a single instrument in a single run. And once the reactions are detected using the optimized EFC recipes from phase 1, the data can be analyzed and the temperatures can be chosen for the next step, which is an isothermal step. So these are data shown for this exact experiment for the MTH1 positive control assay. And keep in mind, this expression plasmid is included in the starter kit, and I highly recommend that this control is run in parallel with experiments for your target of interest. We've already gotten good feedback from the field that clients are using this positive control assay to advantage. And this is an assay that works extremely well in this format. So this is why we include it as the positive control. So here are some features that look very nice. So the black trace is the DMSO control, and you can see that protein denatures as a very steep function of the temperature. And then when a known positive control inhibitor that's potent is included, the TM obviously shifts way to the right, and there's good assay windows, good separation at any given temperature between the signal for the DMSO and compound controls. And so how do you choose the temperatures for the next step? And we outline how to do this in the user manual, but what you want to do is choose the lowest temperatures and then maybe a couple of flanking temperatures that you believe will give you an acceptable assay window. And the reason for that is if you go to super high temperatures, you can impact the apparent affinity of the compound. So the lowest temperature you can go to with an acceptable assay window is what we suggest. So phase 3 assay development is much like the experiments you will actually do once the assay is developed. And these are to measure inhibitor dose response curves at a single temperature. So you're back here and you say, okay, 47 to 50 degrees look like they'd give a good assay window. So let's do dose response curves at those temperatures. So in this case, you would prepare positive control inhibitor, but instead of having it at a single concentration, you would actually make a serial dilution so that you can do a dose response curve. And you'll still harvest the transfected cells, set up the assay, but then what you're going to do is some isothermal denaturation experiments using just a few temperatures that you wanted to bracket based on the data from phase 2. And you'll look at those dose response curves as a function of the temperature and decide which final conditions to use. Good IC50s, good assay windows, and then you know what temperature to lock in on. And once you've developed this very nice and robust assay, you can decide, do I want to make a stable cell line, or do I want to just keep doing the transient transfections, which really aren't that much of a burden. We do them all the time. So here's an example of a successful assay development campaign. Again, I'm using the MTH1 positive control assay as an example. So we identified a temperature, the lowest temperature we could go that gave outstanding assay windows. So here we're actually getting very beautiful assay windows of 4 to 5-fold. We measure accurate rank order potencies for a number of known inhibitors. The negative control, which is a lipid kinase inhibitor, gave no response. And so what we really look for here is when we develop an assay that we would use either as a product or for marketing material, we really want the assay windows to be at least 2, preferably greater than 3. But in practice, assay windows of 2 or maybe even a little less will give very nice dose response curves in this format. The assays have very good, very low noise, so that the curves are fairly robust even with relatively small assay windows. And this MTH1, I forgot to mention, this is, we do offer some assay-ready kits for 2 targets, for MTH1 and then for a kinase I'll talk about in a bit. So if you want to just try out the InCELL Pulse technology before committing to the do-it-yourself starter kit, we provide a kit with a protocol, and it actually includes frozen transfected cells. So those cells can just be plated and used the next day in an assay. It's a very, very convenient format. So now I'd like to get into some of the applications of InCELL Pulse and a case study that just show how we believe this technology can be used, and we really are trying to crowdsource this technology. You folks will all come up with additional applications and utilities for this product, but here's just some examples of how we think it can be used and how we've used it to contribute to the community. So the applications that we believe are going to be of most interest to folks are establishing cellular permeability and target engagement for compounds of interest. So these might be compounds that come from biochemical screens, where obviously the next step is to verify cellular activity. And for an increasing number of targets, I've noticed there just are not boilerplate type easy functional assays available. And so having a solution that's generic like InCELL Pulse is a good way to go to see if your biochemical hits have cellular target engagement potency. This type of technology can also be used to rank order cellular potencies during lead optimization. And I've touched on this earlier, but also analyzing challenging targets where there really isn't a good direct functional assay. Maybe the assay is just cell killing and you'd rather get a little closer to the target. Target engagement assay is very important here, as it is when you're trying to establish the mechanism of action for a compound that was a hit in a phenotypic screen. So you have a hypothesis that you're hitting kinase Y. You better be able to prove that the phenotype you're seeing is due to hitting kinase Y, and if you're getting cellular target engagement, that helps you build the case. And of course, you can use this to de-risk some off-target activities you may see for your compound in broad biochemical screens. And so we've used this technology to validate it across some diverse protein targets, hydrolases, methyltransferases, a whole bunch of different kinases. Kinases is one of our specialties here at KinomeScan, of course. And so just to give you some examples, so ABL1 is a great drug target for leukemia. It's a tyrosine kinase, well-established different types of inhibitors. So there are type 1 inhibitors that bind to the active conformation of ABL, type 2 that bind to an inactive conformation, and there's also non-ATP competitive allosteric inhibitors. We can detect all three types. So for the type 1 and 2, there's dasatinib, which is a highly potent type 1 inhibitor with known cellular activity that's single-digit nanomolar. We're recapitulating that. Imatinib was the first drug approved for this indication, for this target, known to have 100-ish nanomolar activity. We're seeing that. And then this GNF2 compound, this non-ATP competitive compound, binds outside of the active site, but has an allosteric effect by binding to a myristate site that's also part of the kinase domain, and we detect that as well. So that's a very nice feature of this technology, that it can address multiple different sites. And then, of course, a negative control inhibitor showed no activity. And this assay is, just like MTH1, is one of the assays that we provide as an assay-ready kit. If you want to try out the technology, you might have a bunch of known inhibitors. Maybe you're interested in the target in general, but you want to try out the technology before you commit to a campaign of developing your own InCELL Pulse assay using the starter kit. And then, like I referred to earlier, you can use this technology to address targets that are challenging to build functional assays for. So MTH1 hydrolase is a few years back was shown to be an interesting cancer target, and certainly inhibitors had effects on cell proliferation, but it was difficult to build a functional assay. So CETSA was used to prove that the inhibitors of this enzyme were actually cell penetrant and engaging the target. And so we've recapitulated those data very nicely. Like I said earlier, we get the correct rank order potencies. G9A is a methyltransferase, an epigenetic target. Methyltransferases are difficult targets to build functional assays for. The readouts can be complex, oftentimes requiring mass spec. So it's really nice to have a convenient, rapid target engagement type assay to look at the rank order potency of inhibitors, or to show that your inhibitors are actually engaging the target like you think they are. So now I want to finish up with a case study. So DiscoverX is collaborating around kinases with the Structural Genomics Consortium, the SGC. So the SGC has a comprehensive kinase focus at this time, where what they're trying to do is to develop potent and selective chemical probes for every kinase in the kinome. And the reason they want to do that is they realize that there's 500 or so kinases, and only about 15 to 20% of them have dedicated inhibitors and are well understood. There's a potential gold mine of relevant targets for diverse disease indications in the remaining 85%, and chemical probes will pave the way for this initiative. And proof of cellular target engagement for the potent and selective inhibitors that they identify is a key pillar of their strategy. So what do? So what they start with is they have some very nice collaborations with pharma, where they're able to get their hands on some diverse inhibitors, assemble a set, and they screen that set, and they're doing a lot of the screening with us with our Kinome Scan platform, and identify out of these compound libraries that are 500-600 molecules, for which kinase targets do I have potent and selective molecules, with a real focus on the understudied kinases. And so what they ultimately want to do is identify and improve these molecules so that they can test them in phenotypic screens to figure out what kinase targets are good for different diverse disease indications. This is a phenotypic screening approach. It's lengthy, it's expensive, and before they want to start one of those campaigns, they really want to show that they're getting cellular and target engagement against the kinase of interest that they've identified in the biochemical screen. So this is where InCELL Pulse comes in here. So have a collaboration on this with the SGC out of the University of North Carolina, with David Drury, Bill Zerker, and Christopher Asquith, and they've sent us some compounds to test against a couple of the kinase targets for which we've developed InCELL Pulse assays. And these are understudied kinases, GAC and Haspin. This is not VEGFR2, ABL, or MEK. So maybe these kinases are good for disease indications. We've got some biochemical data showing that these compounds are potent and hopefully selective, and now we need the cellular target engagement data. So we were able to successfully run these assays and to generate rank order potency for the series of compounds that were provided. So for the GAC kinase, we've identified very potent compounds, sub 100 nanomolar, and the rank order potencies going from left to right go all the way up to 10 micromolar in a cell, and a similar story for Haspin as well. And so the importance of doing cellular target engagement, especially for something like a kinase, is that not only do you have to worry about cell permeability, but also competition with cofactors. And kinases all bind ATP, which is generally competitive with respect to the inhibitor. And kinases have different affinities for ATP. So it's generally the case that there are 10 to 100-fold offsets between biochemical data and cellular data. So it's important to get these cellular target engagement data to make sure that your compounds are in a relevant potency range in cells, and you know what concentrations to test when you go into the phenotypic screens here, right? You don't get many chances here. These are expensive assays and they take a long time. So you've got to know how to design the experiment so that you won't miss anything. So with that, I'll just finish up by describing a couple of recent posters that have been out on InCELL Pulse, and then we can move into Q&A. So this was an exciting poster for us by Mark Holbert's team at GSK, where they're interested in drug target engagement and have adopted these InCELL Pulse principles and reagents to build some high throughput cellular thermal shift assays for some diverse targets. And so they've shown that this technology is applicable in a higher density format. So most of the kits that we provide, the protocols are for 96 well. A question we get is, can this be moved to 384 or higher? The answer is yes. And then here's some data from a talk and a poster that we gave at Discovery on Target in Boston a couple months back. This poster and the talk featured some of the targets I've already talked about, the hydrolases and methyltransferases, but also featured the fact that we've really gone pretty hard after the human kinome, with the goal of getting assays that broadly cover phylogenetic space. And so we have validated assays now for 25 diverse kinases, tyrosine kinases, serine, threonine, and lipid. And I've mapped these assays onto the phylogenetic tree here. And you can see that for 7 of the 8 major groups, we have at least one representative, and that the distribution is balanced. So we're very excited about applying this technology to kinases and to continue working with the SGC on this. So to summarize, hopefully I've made a case that there is a role for cellular target engagement assays in multiple phases of drug discoveries, independent of the drug discovery paradigm, whether it's phenotypic-based, target-based from the outset. These assays are ideal for establishing that compounds are cell penetrant and get into a cell. And really, I think one of the main value propositions is that the assays have a very simple readout. The the folks in the lab running these assays will like them because it is a very simple luminescent readout without antibody or custom tracer or centrifugation steps. So I think it'll complement the existing tools that are out there very well. In addition to the few assay-ready kits that we provide, our main offering is this detailed starter kit for you to build your own assay, and that includes all the plasmids and reagents you need, as well as a very detailed user manual, which can, like I said earlier, can be downloaded free of charge online, so you can review it and see what you'd be up against for building these assays. And with that, I will stop, and I'd like to thank you very much for your attention, and I'd be happy to address any questions.
Thank you, Dan. That was a nice talk. We have several questions coming in. The first question is, does endogenous expression of the target impact apparent potency reported by InCELL Pulse?
Yeah, so that's a great question, and so if you think about the concentrations of the target in the culture, in the cell culture, and assuming that the compound is freely permeable through the cell membrane, it's difficult to believe that we'll hit the tight binding limit because the endogenous protein is titrating out the compound from the expressed protein. And so looking at the data for very potent compounds like dasatinib, and we've also, for our BTK assay, looked at very potent irreversible inhibitor, we're not hitting the tight binding limit there, even when we're overexpressing the protein. So I'm not worried about titrating the compound with the endogenous protein, and the endogenous protein also shouldn't give any background or anything like that in the assay because it's not labeled with the pro-label tag.
Okay. Okay. So another question was about the system in general relating to full-length versus domains, how well they work for both.
Right.
You might have addressed that. Yeah.
Yeah. So in the talk, I pointed out that the cloning step is critical. So what I've noticed already is that some clients are using whatever construct they have available and generally just sticking it into one of the two expression plasmids. We recommend that you try both the carefully chosen domain-only construct or two, maybe a construct that's used for enzyme activity assays, maybe one from a crystal structure, in addition to the full length. So we do have examples of full-length proteins that work in general for multi-domain proteins where the domains are independently folding. I mean, it's a denaturation-based approach, so ligand binding at one of those domains may not protect other domains in the protein from denaturation aggregation. So we have had success with full-length proteins, particularly in the space of kinases, but I will say that those kinases where we use the full-length construct are generally kinases that have a kinase domain and then not much else on them. So I think this is actually one of the advantages of this engineered system over the SETSA format, where the SETSA format you're dealing with proteins that are expressed as they are in nature, which actually can be looked at as an advantage, but it can bias the technology against multi-domain proteins where ligand binding isn't going to protect the full-length protein from denaturation, so that would be blind to the assay. Here we can use just ligand binding domains that will behave well in these thermal stabilization type approaches and see compound binding. And ABLE is a good example of that. So in the sets of papers I've seen, ABLE kinase is not thermally stabilized by inhibitors, whereas in our engineered system it is, and that's likely because we've lopped off some of the other domains and just focused on the ligand binding domain. So try both.
Okay. Another question came in. Have you tried integral membrane proteins and then maybe address NHRs as well?
Yeah, so no, we have not tried those yet. That's in the plan. nuclear hormone receptors look like proteins that should work very well in this. And also integral membrane proteins. I would have been skeptical about thermal stability type approaches for integral membrane proteins, but a couple of papers came out in the last year or so where CETSA was successfully applied to some diverse integral membrane proteins. Killian Huber from Oxford showed the STING receptor. It's an intracellular membrane protein, works very well by CETSA. And then a number of different integral membrane proteins were shown to work by CETSA in a second paper, and one of those was actually sodium potassium ATP pump. So that was very encouraging, and so we're going to do some internal work on that. But again, I think the value of InCELL Pulse is going to come from the fact that we're trying to crowdsource this technology. We have a very low price for these kits, and I think as a community we can get the most out of this. And I think trying lots of things collectively will really show how far we can go.
Perfect. Okay. There's a question, is a follow-up cell-based activity assay recommended to confirm the engagement? And if so, what do you suggest?
Yeah, so I think that's a great question. So I think just like with biochemical assays, so we have kinome scan here, and we it's a ligand displacement competition binding assay, and we always recommend to clients that if you're getting serious about a compound or a target, to always use an orthogonal assay. So I think cellular target engagement, demonstrating this is probably necessary but not sufficient for some phenotypic change. With functional assays, there's different kinds. So there are coupled assays, there's indirect assays, and then there's very direct assays like autophos for receptor tyrosine kinase, which is about as close to getting cellular target engagement data as you can. But the way I would envision this, doing a drug discovery plan, is if you have a really super robust functional assay that's fairly direct, like something like autophos, where there's not a lot of ways that the assay can fool you, that maybe you don't need something like InCELL Pulse. But in a lot of cases, those types of functional assays don't exist for a lot of these new targets that are popping up. And maybe if they do exist, they're low throughput and cumbersome. So you would build like an InCELL Pulse type target engagement assay as your workhorse assay, and then on select compounds run the orthogonal functional low throughput functional assay every month or so, just to make sure there's good alignment and good agreement between those data. So I think InCELL Pulse is a tool that has to be carefully dovetailed with other tools you have at your disposal when you come up with your drug discovery funnel.
Okay. This is a question related to the format. Is there higher throughput formats available for this? I mean, we provide the 96 well plates, but
yeah, so it can all be adapted to 384 well, and we haven't come up with an exact protocol for that because every lab has different automation, different processes in place. Like I said, the GSK folks have done an amazing job of adopting this technology in 384 well type formats and maybe even beyond. So the core principles don't change when it gets miniaturized, maybe just the instrumentation and liquid handling.
Okay. What is the most important step, during the assay development process, like to not skip? Yeah.
So the data from the thermal melt curve is really your big go, no-go. But from an end user perspective, like anything else in life, the preparation in advance of that is the most important. So think carefully about the cloning. Read the recommendations in the manual. Read some papers on what's worked and what hasn't worked in CETSA, for example. Design your constructs carefully. Don't just use what you already have on the shelf. put the pro-label tag on both sides of the protein. So we've proven to ourselves that that's important. There's a couple of kinase targets that only work in InCELL Pulse if the pro-label tag is on one side or versus the other. And don't skip that very important step at the beginning, the optimization of the readout conditions. You want to make sure that you get a sensitive and true readout of the thermal denaturation of the protein as a function of temperature.
Okay. Final question for today will be, can the target protein expression be adjusted, to more closely match?
Yes, so that's a question that we get a lot, and so folks are, for one reason or another, concerned that overexpressing the protein will have an effect. We got a question already that touched on that in terms of titrating the ligand. People also just feel more comfortable if the expression level is more close to what's normally found in a given cell type. Or, an even more relevant case, in my opinion, is if the protein is part of a higher order complex and has to stoichiometrically interact with other proteins in the cell, you'd like to titrate that down. And so we give recommendations for how to do this in the manual. We validated that this works. And basically, it's very simple. When you do the transfection, dilute the plasmid, the expression plasmid, with the parental plasmid that's empty, that doesn't have an expression construct cloned in, and is just there as basically a decoy. And what that will do is it'll maintain a high transfection efficiency, but a lot of the stuff that's going in doesn't express anything. So you'll get most of the cells transfected, but each cell will express less of the protein. And it's a pretty linear relationship between the amount of protein expressed and the dilution factor you've used of the expression plasmid with the decoy plasmid.
All right. Well, thank you, Dr. Trieber, for your time, and thank you, everyone, for joining the call. As I mentioned at the beginning of the talk, this has been recorded, and you will receive an email to download the recording and slides probably by Friday. All right. Thank you, and have a good afternoon.